Self of Hydra, a presentation by Professor Gershom Zajcek, Faculty of Medicine, Hebrew University of Jerusalem. We are talking about the evolution of life in Gaia. Gaia is a superorganism in which all life evolves and exists, and this is a projection of this Gaia genesis. And in previous presentation, I discussed the evolution of prokaryote, eukaryote, single-cell organism, and now we arrived to the stages of the Hydra, which represent here in a multicellular life. So we are talking about the basic process of viral infection, and which explains all phenomena of this viral infection. Now, virus attaches to a receptor, and its genome is uncoated and injected into the cytoplasm of the cell. Next, the provirus is formed, and the provirus has two tasks. One is it controls the processes in the cell and controls the formation of virions. And the second task is that the set of genes in the provirus stores the self of the cell. And we have seen already in the previous presentation that all life has this self. And it is this very important phenomenon. But here we are interested in viral production. So what happens is that the provirus controls the formation of virions. The virions exit the cell, and the cell dies in lysis. And this process is activated wherever viral infection occurs. So in this Gaia genesis, we are now studying the transition from eukaryotes families into this multicellular unit, and this multicellular unit is called epithelial proliferative unit. And so all epithelial units are the same structure. The basic, you have a stem cell, then there's a progenitor, and finally an end cell. And these cells are progeny of the stem cell, and as the stem cell divides, there's a movement of cell toward the periphery, and this movement I call cell streaming, and the cell differentiate as they move, and once they reach the end cell, they die natural death. So this phenomenon I shall explain in Hydra because it's relatively simple to explain. And here you have Hydra, gut, mouth, and tentacles. Now if we look at this Hydra histologically, we see here the mouth opening, tentacles, and along this wall we have two kinds of units. EPUs. Some units are streaming outwards, other units are streaming inwards. The structure is always the same, and this is the proliferative element in the Hydra. So again, we see that, for instance, the skin unit, we have a stem cell, and cells are streaming outward from the stem cell, and as I said, all these cells are clones of this stem cell, and they differentiate as they go. So here's an example. You have a unit, and the Q cell, the N cell, is removed surgically. So you have a kind of ulcer here, but this ulcer is regenerated by the fact that the unit, or here the progenitors, some of them mature into the end cell, and so you have a regeneration of the EPU, and this is a basic response of a normal or healthy unit. So we are interested in viral infection, and viral infection can occur only in the end cell, because only the end cell has a receptor which can be infected. These cells are they are not mature enough, and they don't have a receptor, and therefore what we are going to talk about will happen only in Q cell. So now we start with the virus which attached to a receptor and infected this Q cell or this end cell, and we got a provirus in the end cell. And now we have the continuation which I described before. The provirus controls the formation of virions. The virions, the cell dies, and the virions are spread into the environment. And as you see, this unit is without end cell, but the end cell will be replaced by the usual turnover of the unit, and we shall have an end cell in the next generation. So, but what is also very important, so we expect that in the new end cell there will be a provirus. This is what we learned also in our previous presentation. But what happens that all units which were infected will have a provirus in their nucleus, although they were not directly infected. So you have here what I described just now. Virus infected this cell and it creates a provirus, but in the next generation of the unit, all the cells got a provirus. So there is an important phenomenon linked with this provirus in these non-infected cells, and it's called endogenous viral element. The provirus in this end is manifested histologically as a kind of as a EVE, endogenous viral element, and this endogenous viral element indicates that all these cells have a provirus, and that all these cells has a provirus with its self. And this is very important. So self stored in the provirus reaches all cells of the EPU, and all the cell in the next generation will have a endogenous viral element. And we shall discuss this phenomenon in future presentation. So back to our virus-provirus process, we realize that this cell can be inspected histologically, because this cell will have a provirus in our unit, and all these cells which have a provirus will have an endogenous viral element, and the endogenous viral element indicates that the provirus has a self. So, short summary, infection starts in end cells, and self stored in the provirus reaches all cells in this EPU, and this happens in all cells which all units which were infected in the end cell, and we shall discuss it. I shall discuss it in future presentation. So, conclusion: viral infection triggers self of all Hydra cells. They all become a family. Without self, homeostasis cannot be maintained, and mind you, these end cells are all in homeostasis. And what we learned in previous presentation is that cell-mediated immunity is an immune response that does not involve antibodies, and this is the manifestation of the EVE, and adaptive immunity is a response that involves antibody, and I shall discuss these responses in future presentation. Enjoy!